Hello students. What happens when we fall sick? Generally, we get fever, sometimes with rigor, and there is pain in the body. So, what your mom will do? She will give paracetamol. Now, paracetamol is analgesic and antipyretic. That is analgesic means it will relieve the pain, and antipyretic means it will lower down the fever. So you feel good for some time, but again the microorganism will grow and it will lead to fever. So how does it happen? Sometimes bacteria or virus, that is antigen, enters our body, and sometimes our WBC soldier cells, they are able to kill immediately and we do not fall sick. But sometimes it takes time, that time we fall sick. So how does these soldiers, they protect our body from the antigen? They produce antibodies. Antigen produces toxin. They produce antitoxin, and they help to protect our body from that antigen. Then WBCs, they are able to come out of the cells. Here you can see how the WBCs they work. They come out of the through these cells, and they are amoebic. That is, they produce a pseudopodia, the false foot, and they come out of the cell of the capillaries, and then they engulf the bacteria and they destroy the bacteria. So here you can see how these cells, the amoebic cells, they come out, they see the antigen bacteria, and how they engulf it. They take inside and they detoxify it. That because antigen that produces toxin, and it is due to the toxin that we get fever. So this bacteria or virus will produce toxin which will lead to the fever. So this is our WBCs, which are going to protect us from all the diseases. So here they are amoebic. They are able to engulf the bacteria. Now, this process of engulfing is known as diapedesis. That is, it oozes out and takes in the bacteria, engulfs the bacteria. So, this process is known as diapedesis. So, here WBCs, they are amoeboid, amoeba-like structure. They produce pseudopodia and squeeze out of the capillaries, and they engulf the bacteria, and that is, they take in the bacteria, destroy it. Now, sometimes the WBCs will be very high in number. So that's a disease which is known as leukemia. Leukemia is a blood cancer, and only blood transfusion helps. Now, the other, when the WBCs are very less, leukopenia, now that can be due to some problem, and that disease can be treated. So this is what happens with the WBCs. Now let us see what is the classification and how the WBCs they work. So here, in general, the lifespan is for 2 weeks. And this WBCs, they are also known as leukocytes. Their number is from 4,000 to 8,000 per mm cube. That is, it is volume. In that 1 mm volume, you can see 4,000 to 8,000 WBCs. Now, if it goes to 50,000, that means your body is fighting an infection. So, whenever you have fever and get your blood test done, that time your WBCs will be very high. That means your body is fighting an infection. Now, in general, All the WBCs are for protecting our body, but they have some classification and particular job to do. Now, there are two categories of WBCs. One is granular and one is non-granular. Naturally, when granules are present in the cell, then granular. Present in the cytoplasm of the cell, then it is granular. Cytoplasm contains granules. Nucleus usually constricted into lobes. Lobes are formed in the nucleus. That is, nucleus is not one solid thing, but it is in the form of lobes. The other is non-granular. That means the cytoplasm will not have any granules. They have single large nucleus. There's only one single nucleus. What are the types of WBCs? Now, WBCs are white in color. You cannot see under microscope properly because there's no color. So they will be generally given some stain. They will be colored and then you will be able to see the WBCs. Now, if they take acidic stain, it is eosinophil. If it takes basic stain, then it is basophil. And if it takes neutral stain, then it is neutrophils. So here types neutrophils. They are large in number, 55 to 70% are neutrophils. They are lobed. You can see this nucleus, it is lobed. It might be 3 or 4 lobes. Nucleus, it is in form of 3 or 4 lobes. Stains with neutral dyes. It is therefore neutrophils. It engulfs the bacteria. It engulfs. It takes in the bacteria and whatever toxin it detoxifies. So it is phagocytic. It is amoebic. It takes out its pseudopodia, engulfs it. Therefore, this process of engulfing is known as phagocytosis. It is taken in the cytoplasm and detoxified. It engulfs the bacteria by the process of phagocytosis, engulfs and detoxify. Now they are produced in the bone marrow. All these cells are produced in the bone marrow, in the red bone marrow. Yellow is for storing. Yellow bone marrow is for storing the fats. Red bone marrow produces this WBCs. Now, this neutrophils, they remain only for few hours, and 125 billions that is produced every day. Every day, 125 billion is produced. So many are produced for protecting our body. Now, next is eosinophil. That comes to 1 to 3%. And during the time of allergy, now your body sometimes might react to some useful substance or substance which are not harmful, and you develop allergy, mucus, all those problems you develop because of eosinophil. The count increases in allergies. It is 1 to 3%. Now nucleus here is two lobed. Stains dark red with eosine, and therefore the eosine is the acidic dye and acidic stain, and these cells are known as eosinophils. Now this eosinophil also engulfs the bacteria and it secretes antitoxin. The allergy might be due to some substance which is toxic to the body. Therefore, eosinophil will produce antitoxin to detoxify. It is associated with allergy. It is produced in red bone marrow. Now, basophil, they are 0.5 to 1%. Here, the nucleus is not distinctively lobed, but still it is lobed. Nucleus large and indistinctly lobed. granular stain with basic dyes. So basophil because it has taken basic dye. Example, methylene blue. It takes the stain of methylene blue. It releases histamine. It's a chemical which is produced due to the allergy and causes inflammation. Inflammation, it will be due to some toxic, it will inflate. So inflammation will be there. It dilates the blood vessel during inflammation. Now supposing there is a cut. There is a cut. What will happen? After some time, pus will be formed. Why? Because there are dead tissues, dead WBCs, and the bacteria which has entered to this punctured skin. So here you will see the pus. So pus is the WBCs, dead tissues, and the bacteria. So whenever there is some rupture, inflammation will be there. So this helps to dilate the blood vessel. What? Histamine. It helps to dilate the blood vessel during inflammation. Now bone marrow, red bone marrow, it produces basophils. All these cells are produced in the red bone marrow. Now this is granular. These are granular WBCs. They have granules in the cytoplasm. Now the cytoplasm, when it does not, now here in the neutrophils the granules are small in size, whereas the granules in eosinophils, they are larger in size. So that is the difference between the granules. Now, non-granular means no granules present in the cytoplasm. Cytoplasm without granules, single large nucleus. Now, lymphocytes. Lymphocytes are produced in the lymph nodes also. So they consist of 20 to 35% of the total WBCs. Here they have single large nucleus. They are the smallest in size, smallest of WBCs. They have single large nucleus. They produce antibodies against antigen. Now, whenever the microorganism enters, it produces bacteria or virus, that's antigen. Our body produces antibodies to kill it. It is toxin. So this produces antitoxin to detoxify it. So this is antibodies are antitoxin. Antigen is toxin. So antibodies are produced to detoxify this antigen. So antitoxin is produced for toxin. So this is how a body protects. But supposing a virus or bacteria enters our body, that is antigen. Our body produces antibodies to kill it, to detoxify it. So now these antibodies our body will produce throughout. So if you have suffered from any disease in childhood, you will not suffer in adulthood because those antibodies are already present in your body. So this principle is used for vaccination. While vaccination is given, this antigen is introduced, weakened antigen is introduced, which cannot cause harm. There will be slight increase in temperature, but it won't be able to cause harm. But the body will be immune to that disease. So in childhood, polio vaccination is given. When a polio virus enters the body of the child, it will cause fever and the legs or limb will be paralyzed. So fever will go away and antibodies will be produced, but the limbs will be damaged for life. So immunization is given. So actually when the viruses, they enter the body, already antibodies are there. They will detoxify the antigen. They won't cause any harm to the body. So this is how vaccination works for all the diseases. Why? Because antigen is introduced before weakened antigen, the weakened microorganism are produced, so antigen will enter, our bodies will produce antibodies, and when the actual antigen enters, antibodies are there to fight it. Antigen and antibodies are very specific. Supposing this is the antigen, shape of the antigen, antibodies will be like this to detoxify, to nullify it. So now we already have this type of antibodies for this type of antigen. So actual antigen, when it enters, our antibodies will not let it cause any harm. So it produces antibodies against antigen. Very important lymphocytes. Now next is monocytes. Largest of the RBCs. This is smallest of the RBCs. Here it is largest of the RBCs. Nucleus large. It is kidney shaped. Nucleus is in kidney shape. It transforms into macrophages at the site of infection. Supposing there's an infection on the skin, it will turn into macrophage. That is, it will engulf the bacteria which has entered. So wherever it is required, it will turn into macrophage and engulf the bacteria or whatever the infection. It ingests the germs. So macrophage, this monocytes, they turn macrophage and ingest the germs which has entered the body. They are also produced in the red bone marrow. So all these are produced in red bone marrow. But this lymphocytes, along with the red bone marrow, they are also produced in the lymph glands. Which are the lymph glands? Spleen and tonsils and etc. Where lymph glands, they produce lymphocytes. So these are the cells. They are the WBCs. They are the cellular part of the blood. RBCs, they are red in color. They have hemoglobin. WBCs, they are colorless. They do not have hemoglobin. RBCs, they do not have nucleus, but WBCs, they have nucleus. So protecting our body is the main function of the WBCs. They do not allow the microorganism to affect us. They are continuously fighting. But if supposing we get infected due to some reason, our WBCs are not strong enough, then still they will go, after entering, will suffer from high temperature, all those things. But then our cells are still fighting. They are trying to produce the antibodies for that antigen, and they kill that microorganism and protect us. This is how our WBCs, they work to protect our body. So here we should develop our immunity system. How we can do? By having good nutrition. So good proteinaceous nutrition helps us to build a better immunity system. So these soldiers are strong, and then they can protect our body. But if we are fond of junk food which does not consist of protein and only sugar and not all the nutrient, then you will have weak immunity, and these cells are not so developed to protect you. So you yourself have to build the immunity.